Hey guys, can everyone see this PowerPoint? Okay, perfect. Awesome. As Mickey said, my name is Keshav. I'm a senior double majoring in mole cell and math. And I'm here to talk a bit about my research for the past couple of years in elucidating novel determinants of resistance to antibiotics and mycobacterium. So before I begin, I just wanted to provide a bit of background to help you guys understand a bit more about the work that my lab and I have been doing. So essentially what we do is we basically evaluate the efficacy of novel drugs and drug regimens against tuberculosis using in vitro and in vivo models. Scientists and physicians in the past, couple of decades have realized that there's a tremendous need for a couple of things. Number one, there's a tremendous need for new drugs against tuberculosis. As multi-drug resistant, extremely drug resistant tuberculosis is becoming more and more common, all the drugs that we've had in the past aren't really working as well. And so we've had to resort to using these second and third line therapies that are a lot more toxic to the human body and that require the administration of these drugs from anywhere from 9 months to at most 2 years. And obviously that's not ideal when you're trying to treat, any type of illness because of the probability that patients won't complete the entire course of regimen, therefore leading to more antibiotic resistant bacteria. And additionally, there's a need for new detection methods in the sense that only 1/3 of multi-drug resistant cases are estimated to be properly identified upon the initial diagnosis. And obviously, if you don't accurately identify the strain of multi-drug resistant tuberculosis present in a patient, you can't know what the resistance profile of that strain will be. And any non-ideal therapies you use to try to treat that infection can then create more antibiotic resistant drugs. So therefore, there's also a need to better identify, what type of strain and what the resistance profiles of that strain are so that we can give the most effective therapies to cure that patient. So we've been kind of lucky. So in the past 50 years, only 2 new classes of drugs have been approved to treat tuberculosis. And one of these is called the bicyclic nitroimidazoles. And basically what this new class of drugs does is that it actively inhibits both non-replicating and rapidly growing bacteria in both aerobic and anaerobic conditions. So it's really this really novel, super effective new type of drug that is able to treat a lot of, cases of tuberculosis that we were having a difficult time treating with currently existing therapies. My lab primarily works with 2 of these new bicyclic nitroimidazoles. On the left, you can see the chemical structure of protamonid. This is the newer one that was approved around 2019 for use in patients. And we also work with delaminid, which is a slightly older drug with a little bit less clinical relevance, but it still is effective in combination with other therapies. And both of these drugs are initially present in the prodrug form. So they're not immediately activated as soon as you ingest them. They need a bit of, there's a bioactivation mechanism involved in activating these drugs. So without getting too much into the weeds of exactly how these drugs are activated, basically a pathway called the F420 biosynthesis pathway creates a cofactor called F420. There's another protein in the cell, in the Mycobacteria cell. It's the only one known to exist that reduces this cofactor F420. And then another molecule, DDN, then oxidizes F420. So it becomes, it's not in this reduced form anymore. But what DDN does is it's able to transfer that hydride from the reduced form of F420 to the prodrug versions of both protamonid and delaminid, and that allows it to be activated. So this pathway is essential to activating protamonid and delaminid and helping it, develop these antimicrobial properties when it's ingested and metabolized in the body. So just going a bit more into the F420 biosynthesis pathway. This is a non-essential pathway that helps Mycobacterium tuberculosis fight against oxidative stress. And recently in 2019, scientists think that they have, identified what this pathway exactly looks like. So first, as you can see up here, the molecules themselves aren't that important, but what's important to note is that the FO synthesis, FBIC, this is the determining factor, the determining enzyme that basically launches this pathway and makes it irreversible. So it is responsible for creating a precursor to F420 called FO. Another molecule then, FBIA, then combines a phosphoenolpyruvyl moiety to this FO to create yet another intermediate step in the F420 biosynthesis pathway. And then finally, FBIB then reduces this intermediate, and then just does a couple other things to finally make it into F420. And recently, these three enzymes were thought to be the main enzymes in this pathway. But Bashiri et al. In 2019 discovered that there may be a third drug called FBID that basically makes this phosphoenolpyruvyl moiety and is essential to this entire pathway. So again, like I said before, because this pathway is required to activate both protominid and delaminid in the body, mutations in any gene in this pathway, as well as in FGD and DDN, which are also required to activate protominid and delaminid, mutations in any 6 of those genes could potentially, cause resistance to protominid and delaminid in Mycobacterium tuberculosis. And then so that's what my lab sought to figure out. So the main project that I'm going to present in this presentation is something that we actually just had published in Antimicrobial Agents and Chemotherapy. The preprint version is accessible in a link I provide at the end of this presentation. But basically, we found that mutations in this new gene, FBID, is a novel determinant of resistance to protominid and delaminid in Mycobacterium tuberculosis. So basically, as a bit of background, what we saw is that, we were motivated for this study because there was a lack of data concerning the evolution of in vivo resistance during treatment with protominid and delaminid. And this is especially concerning because prior experiments have shown that compared to other anti-tubercular drugs, both of these nitroimidazoles don't really have that big of a barrier to resistance. Yeah, thanks for the congrats. Yeah. So basically, what we found is this is like the motivation for the study. And then so we just sought to see if we could identify a certain grouping of genes that if we detect mutations in any of those genes, we can say, okay, this particular clinical strain may be resistant to nitroimidazoles, and therefore let's combine it with these other drugs. So that was the motivation going into the study. My role in this study was basically performing the MIC studies. So essentially, before I came on, our lab selected and sequenced mutants that were resistant to protominid. And then I conducted MSc studies to see the extent to which these mutations conferred resistance to both protominid and delaminid. And so what that means is I'm trying to basically see how much more resistant the mutants got to these new nitroimidazoles after they've had their mutation. And previous literature indicated that because both of these drugs were from the same class of antibiotics, one mutation, if it conferred resistance to one of these drugs, it should also confer resistance to another drug. And I'll get a bit more to my results later, but that really wasn't the case. But back to the MSCs themselves. So basically how I did this was using a broth microdilution assay. And what that essentially means is that I first inoculated bacteria so that they grew to about 10 to the 5th CFU per milliliter in a particular broth that we use to grow mycobacteria. I then added varying amounts of protominid and delaminid to each of these test tubes, and I did this experiment in triplicates. And then finally, I let these test tubes incubate for 14 days at 37 degrees, and then I tried to see if there was any visible growth of mycobacteria in any of these test tubes. Because essentially what an MIC is, it's an acronym for the minimum inhibitory concentration, and that essentially is the lowest concentration at which no visible bacterial growth is present. And so using this methodology, what I found is pretty much summarized in this table here. The first row refers to the MICs of protominid and delaminid in wild type mycobacteria. So you can see those values right there. And it seems that, for the vast majority of these genes, mutations in one gene, for example, gives you tremendous resistance to both protominid and delaminid. If you see on the bottom of the table, for example, like certain mutations in the genes increased the resistance of the bacteria to pertaminid by like a factor of 128, and the same with delaminid. But my results did show two really interesting concepts. And it was the fact that the genes in the box region that I have, FBID and FBIB, those mutations resulted in a tremendous increase to pertaminid resistance, but not to delaminid resistance. So if you can, take a closer look at that table, we basically see 128 factor of increase for resistance against pertaminid for these mutants, but the resistance to delaminid only increased by a factor of about 8 to 12, 8 to 16. And obviously that seems kind of odd, why do we see this tremendously large increase in pertaminid MIC, but not for the delaminid MIC? And that's something that we're still trying to study. But the main factor that I did want to point out is that mutations in FBIB and FBID only conferred low-level resistance to delaminid and not, other genes. And that was kind of confusing. We still don't know what why this differential impact of FBIB and FBID, exists. We have an idea, our lab has a theory that it could be like the chemical structure of these two drugs. For example, we know that because as you saw in like one of the background slides, like the structures are slightly different. So perhaps they interact with the intermediates in different ways, or perhaps, delaminid requires less of the intermediate than pertaminid in order to become fully activated. And that's why the resistance isn't as much, but we still don't know. We still don't know exactly why, and that's the subject for future discussions. But the important part was that we found out that through my experiments and experiences in my lab, that these two genes, which we previously thought would confer resistance to both pertaminid and delaminid, only conferred high-level resistance to pertaminid. And that's extremely important when we're trying to figure out these new novel regimens that we can use to tackle some of these clinical strains that are resistant to one antibiotic over the other. Just some other pertinent findings from our study. We did indeed establish that FBID is required for F420 biosynthesis, and we also determined that it was a novel determinant of nitramidazole resistance. Previous literature has suggested that the other 5 genes that I mentioned, the 3 previously known genes in the F420 biosynthesis pathway, as well as the reductase and the oxidate that reduced and oxidized F420, they already knew that those 5 genes, if they had mutations in them, could yield resistance against nitroimidazoles. But the 6th gene that we tested was a new gene that we didn't know before if it was a determinant of nitroimidazole resistance or not. So again, together, those 6 genes we can safely say comprise a set of non-essential targets for spontaneous resistance of unprecedented size for a tuberculosis drug. In our study, I think we had about 150 different mutations in all 6 of these genes, and pretty much I think 70% of those mutations weren't doubled or tripled, which means that they were all unique. So most drugs don't have this wide of a target range, but it seems from our study that although these nitroimidazoles are very effective at combating some of these clinical strains that don't respond to other treatments, they still have a humongous target area for mutations that could potentially confer resistance to them. And of particular interest, and what motivated my previous experiments, my future experiments, was that pertaminid-resistant FBID mutants were hypersensitive to other tuberculosis drugs. And so what that means is that even if these mutants were resistant to pertaminid and delaminid, they were extra sensitive to other tuberculosis drugs. So ideally, if we could then combine that pertaminid with some other drugs that the mutant was hypersensitive to, that could then create a very effective cocktail of drugs used to fight these multi-drug-resistant clinical strains that exist today. So just some quick future directions. Last year, I basically helped my lab create a transposon mutant library. And so what that essentially is just a library of mutants with a bunch of coverage that have transposon insertions in not all non-essential genes. And that basically means that we have this library that has pretty much every single knockout that you could dream of that still allows bacteria to live. And the ultimate purpose of this library is that it allows us to expose it to an array of drugs to determine, again, novel determinants of resistance, because that's what we want to find out. And I kind of applied this to an independent project that I'm studying currently, a tetracycline comparative study. So tetracyclines are another classification of antibiotics that have been used to treat tuberculosis. Two particular members in tetracyclines are tigecycline and amatocycline. Tigecycline, I think, was approved for TB in like 2004, so it's kind of old. It's an older generation antibiotic. Amatocycline was recently, approved. I think 2018, 2019, somewhere around there. So it is a newer generation analog of tigecycline. And basically, what I'm trying to do is expose that transposon mutant library that I was talking about in the previous slide to these drugs, and then hopefully identify mutants that are resistant to either one of these drugs or both. And then by identifying those mutants and then sequencing them, we can see which genes confer resistance to both of these drugs. And then we can also then further see, in years later, if those genes also confer hypersusceptibility to other tuberculosis drugs. And if they do, combining these medicines with other drugs to create more effective cocktails. Again, because of the whole coronavirus pandemic, I wasn't able to finish this project, so I'm still, it's still ongoing, but I aim to continue and finish it after restrictions are being lifted. So yeah, that's a bit about the research that I've been conducting for the past 3 years. I have my references right here, and I'd love to open up the floor to any questions you guys may have.
All right, any questions for Keshav? I totally feel you on the research thing. Like I felt like I was just hitting my stride and then they were like, all right, go home.
Yeah, no, straight up, like, I think literally 2 weeks before I was gonna isolate my samples, they sent us home. And if I had just isolated the DNA from those samples, I could have just sent it on to sequencing, and then we could have like done all the data analysis from home. But
oh, what a bummer.
Yeah, it was unfortunate, but yeah, the preprint made up for it. So
there you go.
I'm
There you
excited
go.
about that.
All right. Any questions? I'll give it a couple of beats. All right. Excellent job presenting.